Lab Protocols and Solutions. Assalamu alaikum everyone. Today we will discuss laboratory protocols and solutions, which are among the most fundamental concepts in every scientific laboratory. Whether you are working in microbiology, biochemistry, pathology, biotechnology, pharmaceutical sciences, or molecular biology, following proper laboratory protocols and preparing solutions accurately are essential for obtaining reliable and reproducible experimental results. A laboratory protocol is a written set of standardized instructions that guide scientists through every step of an experiment. These protocols ensure that all procedures are performed consistently regardless of who performs them. Standardization reduces errors, improves reproducibility, and enhances laboratory safety. Protocols are particularly important because many laboratory experiments involve hazardous chemicals, infectious microorganisms, or expensive instruments. Even a small mistake can lead to inaccurate results, contamination, equipment damage, or accidents. Another important topic is laboratory solutions. A solution is a homogeneous mixture consisting of a solute dissolved in a solvent. Laboratory solutions are used in almost every experiment, including preparing culture media, staining tissues, extracting DNA, performing biochemical assays, and calibrating instruments. Proper preparation of solutions requires accurate weighing, measuring, mixing, labeling, and storage. Any mistake in concentration can significantly affect experimental outcomes. By the end of this lecture, you will understand laboratory protocols, safety rules, solution preparation, dilution techniques, molarity calculations, and good laboratory practices. Key points. Importance of standardized procedures. Accuracy in scientific experiments. Laboratory safety. Quality control. Reproducibility. Learning objectives. The learning objectives describe what students should be able to understand after completing this lecture. The first objective is to understand laboratory protocols. Students should know what protocols are, why they are important, and how they contribute to scientific accuracy. The second objective is to prepare laboratory solutions correctly. This includes understanding stock solutions, working solutions, standard solutions, buffers, concentration units, and dilution calculations. The third objective is to follow laboratory safety guidelines. Every laboratory worker must understand the importance of personal protective equipment, PPE, chemical safety, biological safety, waste disposal, and emergency procedures. Students should also develop practical laboratory skills, including accurate measurement, pipetting, weighing chemicals, labeling containers, recording observations, and maintaining laboratory notebooks. Finally, students should appreciate the importance of quality assurance and good laboratory practices in producing reliable scientific data. Learning outcomes. Explain laboratory protocols. Prepare different laboratory solutions. Calculate concentrations. Perform dilution calculations. Follow laboratory safety rules. Maintain accurate laboratory records. What is a laboratory protocol? A laboratory protocol is a written document containing detailed step-by-step instructions for performing an experiment or laboratory procedure. Protocols ensure that every researcher performs the experiment in exactly the same way. This minimizes variation and improves reproducibility. A protocol usually contains objective, required materials, equipment, chemicals, safety precautions, experimental procedure, data recording, expected results, waste disposal instructions. For example, a DNA extraction protocol specifies exactly how much buffer to add, incubation time, centrifuge speed, washing steps, and storage conditions. If one scientist changes any step, the results may become unreliable. Protocols also improve laboratory efficiency because new staff can quickly learn procedures by following written instructions. Laboratory protocols are often called standard operating procedures, SOPs. Advantages. Standardization. Accuracy. Safety. reproducibility, reduced human error, better quality control. Laboratory safety. Laboratory safety is the most important aspect of scientific work. No experiment is successful if safety rules are ignored. Every laboratory worker should wear appropriate personal protective equipment, PPE, including a lab coat, gloves, safety goggles, and closed shoes. PPE protects against chemical spills, biological hazards, and physical injuries. Eating, drinking, or using mobile phones in laboratories is prohibited because these activities increase the risk of contamination. Students should know the location of emergency equipment such as fire extinguishers, first aid kits, eye wash stations, emergency showers, spill kits, and emergency exits. Chemical containers should always be labeled properly, and safety data sheets, SDS, should be available for hazardous substances. Waste must be segregated into chemical, biological, sharps, and general waste containers. Always report accidents immediately to the laboratory supervisor. Good housekeeping, proper ventilation, and maintaining a clean workspace reduce laboratory accidents. Remember: safety first, experiment second. Laboratory equipment. Laboratory equipment enables scientists to perform accurate experiments. A beaker is used for mixing, heating, and holding liquids. It provides approximate volume measurements. A flask, especially a conical, Erlenmeyer flask, is used for mixing solutions without spilling and is suitable for titration. A graduated cylinder measures liquid volumes more accurately than a beaker. A pipette transfers small liquid volumes with high precision. A micropipette measures very small volumes, typically between 0.1 L and 1000 L, and is widely used in molecular biology, microbiology, and biochemistry. Other important equipment includes analytical balances, centrifuges, pH meters, spectrophotometers, incubators, autoclaves, hot plates, and magnetic stirrers. Proper calibration and maintenance of equipment are essential for obtaining accurate results. Always clean equipment after use and store it according to laboratory guidelines. Common Viva questions. Difference between a pipette and a micropipette? Why is calibration important? Which instrument measures pH? Which equipment measures mass accurately? Types of solutions. In every laboratory, solutions are used in almost every experiment. A solution is a homogeneous mixture in which one substance, the solute, is completely dissolved in another substance, the solvent. Understanding the different types of laboratory solutions is essential for preparing accurate reagents and obtaining reliable experimental results. 1. Stock solution. A stock solution is a highly concentrated solution prepared in advance. Instead of making a fresh solution every time, scientists prepare a concentrated stock and dilute it whenever needed. This saves time, reduces chemical waste, and ensures consistency between experiments. Example: a laboratory may prepare a 10 m NaCl stock solution. When a 1 m solution is needed, it is prepared by dilution. 2. Working solution. A working solution is the diluted form of the stock solution and is used directly in experiments. Working solutions should be prepared carefully because incorrect dilution can affect experimental outcomes. Example: a PCR buffer diluted from a stock solution before use. 3. Standard solution. A standard solution has a precisely known concentration. It is mainly used for titration, calibration of laboratory instruments, and analytical chemistry. Standard solutions are prepared using analytical-grade chemicals and accurate volumetric glassware. Example: 0.1 m hydrochloric acid, HCl. 4. Buffer solution. A buffer solution resists changes in pH when small amounts of acids or bases are added. Buffers are essential in biological experiments because enzymes and cells require a stable pH to function properly. Examples: phosphate buffered saline, PBS. Tris buffer. Acetate buffer. Importance: ensures experimental consistency. Maintains chemical stability. Improves reproducibility. Reduces experimental errors. Practical example. If preparing an ELISA test, the washing buffer must have the correct concentration and pH. An incorrect buffer may produce false positive or false negative results. Exam tip. Remember. Stock concentrated. Working used in experiment. Standard known concentration. Buffer maintains pH. Solution components. Every laboratory solution consists of two basic components. Solute. Solvent. Solute. The solute is the substance that dissolves in another substance. Examples include sodium chloride, NaCl, glucose, copper sulfate, potassium chloride. Solvent. The solvent is the substance that dissolves the solute. In biological and chemical laboratories, distilled water or deionized water is the most commonly used solvent because it is free from impurities. Examples: water, ethanol, methanol, acetone. Solution. When the solute dissolves completely in the solvent, a homogeneous mixture called a solution is formed. Example: 10 g NaCl plus 100 ml distilled water equals salt solution. Factors affecting dissolution: temperature, stirring, particle size, nature of the solute, nature of the solvent. Increasing temperature generally increases the solubility of most solids. Stirring speeds up the dissolution process by increasing contact between particles. Types of solutions: unsaturated solution, saturated solution, supersaturated solution. Laboratory applications. Solutions are used in biochemistry, histology, hematology, molecular biology, microbiology, pharmaceutical manufacturing. Practical example. To prepare 0.9% normal saline. Weigh 9 g NaCl. Add distilled water. Make the final volume to 1 liter. Mix thoroughly. Label and store properly. Key concept. Solute plus solvent equals solution. Concentration. Concentration describes the amount of solute dissolved in a specific amount of solvent or solution. It is one of the most important concepts in laboratory science because the concentration of a solution directly affects experimental results. Common units of concentration. Percentage, percent symbol. Expressed as grams per 100 milliliters. Example. 5% glucose means 5 g glucose in 100 milliliters solution. Molarity, m. Moles of solute per liter of solution. Formula. M equals moles/liters. Normality, n. Number of gram equivalents per liter of solution. Used mainly in acid-base titrations. Parts per million, ppm. Used for very dilute solutions. 1 ppm equals 1 mg per liter. Why concentration matters. Incorrect concentration can kill cultured cells. Produce false laboratory results. Damage sensitive equipment. Reduce enzyme activity. Practical example. Preparing disinfectant. If bleach is too concentrated, it can damage equipment. If too dilute, it may not kill microorganisms effectively. Tips. Always. Use analytical balances. Measure accurately. Read the meniscus correctly. Mix thoroughly. Label the solution. Molarity. Molarity is one of the most commonly used methods of expressing concentration in chemistry and biology. Definition. Molarity is the number of moles of solute dissolved in 1 liter of solution. Formula. M equals moles of solute/volume, liters. What is a mole? A mole is 6.022 times 10 superscript 2 superscript 3 particles, Avogadro's number. It allows scientists to measure the amount of a substance based on the number of molecules or atoms. Example: molecular weight of NaCl equals 58.44 grams per mole. To prepare 1 m NaCl, weigh 58.44 g NaCl. Dissolve in distilled water. Make the volume up to 1 liter. Mix well. Label the bottle. Applications: buffer preparation, cell culture media, PCR reagents, biochemical assays, drug preparation. Common mistakes: incorrect molecular weight, wrong final volume, poor mixing, measurement errors. Viva questions: what is molarity? What is 1 mole? Why is molarity preferred in laboratories? Preparing standard solutions. Preparing a standard solution requires precision because the concentration must be exactly known. Standard solutions are widely used in analytical chemistry, titration, instrument calibration, and quality control. Step 1. Calculate the required amount. Use the required concentration and molecular weight to calculate how much chemical is needed. Step 2. Weigh the chemical. Use an analytical balance for maximum accuracy. Never estimate the amount. Step 3. Dissolve the solute. Transfer the weighed chemical into a beaker and add a small amount of distilled water. Stir until it dissolves completely. Step 4. Transfer to a volumetric flask. Carefully pour the solution into a volumetric flask using a funnel. Rinse the beaker to transfer all the chemical. Step 5. Make up the final volume. Add distilled water until the bottom of the meniscus reaches the calibration mark. Step 6. Mix thoroughly. Close the flask and invert it several times to ensure uniform mixing. Step 7. Label the solution. Include solution name, concentration, preparation date, expiry date, prepared by, hazard information if applicable. Step 8. Storage. Store under the recommended conditions, such as room temperature, refrigeration, or protection from light. Quality control. Before use, check for contamination. Verify the label. Inspect for precipitation or color changes. Confirm the expiry date. Practical example. To prepare 500 milliliters of 0.1 m NaCl. 1. Calculate the required mass. 2. Weigh the NaCl accurately. 3. Dissolve it in distilled water. 4. Transfer to a 500 milliliters volumetric flask. 5. Make up the volume to 500 milliliters. 6. Mix, label, and store correctly. Dilution. Dilution is one of the most frequently performed procedures in laboratory work. It involves reducing the concentration of a solution by adding more solvent, usually distilled or deionized water. Dilution allows scientists to prepare working solutions from concentrated stock solutions while maintaining accuracy and consistency. The principle behind dilution is that the amount of solute remains constant, while only the volume of the solution changes. This concept is represented by the dilution equation. CD equals C₂V₂. Where C equals initial concentration, stock solution. D equals volume of stock solution required. C2 equals final concentration, desired concentration. D2 equals final volume after dilution. Example calculation. Suppose you have a 5 m NaCl stock solution, and you need to prepare 100 milliliters of a 1 m solution. Using the formula CD equals C₂V₂. 5 times V is equal to 1 times 100. D equals 20 milliliters. Take 20 milliliters of the stock solution and add distilled water until the total volume reaches 100 milliliters. Why dilution is important? Dilution is necessary because concentrated solutions are often too strong for direct use, hazardous to handle, expensive to prepare repeatedly, Unsuitable for biological samples. Preparing working solutions through dilution saves time and ensures consistent results. Types of dilution. 1. Simple dilution. One-step dilution from stock solution. 2. Serial dilution. A sequence of repeated dilutions, commonly used in microbiology, immunology, and molecular biology. Example. 110. 1:100. 1:1000. 1:10,000. Serial dilution is widely used to determine bacterial concentration, antibody titers, and drug potency. Laboratory applications. ELISA. PCR reagent preparation. Microbiology cultures. Clinical biochemistry. Drug preparation. Cell culture. Common mistakes. Using the wrong stock concentration. Measuring incorrect volumes. Forgetting to mix thoroughly. Contaminating the stock solution. Incorrect calculations. Safety tips. Always use calibrated pipettes. Wear PPE. Use clean glassware. Label diluted solutions immediately. Viva questions. What is dilution? State the dilution formula. What is serial dilution? Why is dilution important? Key takeaway. Dilution changes concentration but does not change the amount of solute. Buffer solutions. Buffer solutions are special solutions that resist changes in pH when small amounts of acid or alkali are added. Buffers are extremely important because most biological reactions occur only within a narrow pH range. If the pH changes significantly, proteins may denature, enzymes may stop working, and cells may die. Components of a buffer. A buffer contains a weak acid and its salt, or a weak base and its salt. These components neutralize added acids or bases, preventing sudden pH changes. Common laboratory buffers. Phosphate buffered saline, PBS. pH approximately 7.4. Used for washing cells and tissues. Tris buffer. Common in molecular biology. Used in DNA and protein studies. Acetate buffer. Used in acidic pH experiments. Functions of buffers. Maintain stable pH. Protect enzymes. Preserve proteins. Maintain cell viability. Improve experimental reproducibility. Applications. Buffers are used in PCR. DNA extraction. Protein electrophoresis. ELISA. Histology. Cell culture. Biochemistry. Factors affecting buffer efficiency. Temperature. Concentration. Buffer capacity. Correct pH adjustment. Preparation of buffer. 1. Weigh chemicals accurately. 2. Dissolve in distilled water. 3. Measure pH using a calibrated pH meter. 4. Adjust pH using HCl or NaOH. 5. Make up the final volume. 6. Label and store. Common mistakes. Incorrect pH adjustment. Using expired chemicals. Poor calibration of pH meter. Contamination. Viva questions. What is a buffer? Why is PBS commonly used? Name 2 laboratory buffers. What happens if pH changes during an experiment? Key point. Buffers protect biological systems by maintaining a stable pH. pH measurement. The pH of a solution indicates its acidity or alkalinity. It is one of the most important parameters measured in laboratories because many chemical reactions and biological processes depend on a specific pH range. pH scale. pH 0 to 6 acidic. pH 7 neutral. pH 8 to 14 alkaline, basic. Methods of measuring pH. 1. pH paper. A strip of indicator paper changes color depending on the pH of the solution. Advantages: quick, cheap, easy to use. Disadvantages: less accurate. 2. pH meter. A pH meter uses an electronic electrode to provide precise pH readings. Advantages: highly accurate, suitable for research laboratories. Calibration of pH meter. Calibration should be performed daily before use. Common calibration buffers: pH 4, pH 7, pH 10. Calibration ensures accurate measurements. Factors affecting pH measurement: dirty electrode, temperature, improper calibration, air bubbles, expired buffers. Applications: water analysis, food industry, pharmaceutical manufacturing, cell culture, clinical laboratories, environmental monitoring. Proper procedure: 1. Switch on the pH meter. 2. Rinse electrode with distilled water. 3. Dry gently with tissue. 4. Immerse electrode. 5. Wait until reading stabilizes. 6. Record pH. 7. Rinse electrode again. Viva questions. Define pH. Which instrument measures pH accurately? Why is calibration necessary? Key point: a calibrated pH meter provides accurate and reliable pH measurements. Labeling. Proper labeling is one of the simplest yet most important laboratory practices. Incorrect or missing labels can lead to sample mix-ups, contamination, incorrect experiments, and even serious laboratory accidents. Every prepared solution and sample must be labeled immediately after preparation. Information on a laboratory label: name of solution, concentration, date of preparation, expiry date, prepared by, storage conditions, hazard symbols if applicable. Example label: name, sodium chloride solution, concentration 0.9%, prepared July 5, 2026, expiry August 5, 2026. Prepared by Masroor Ahmed. Store at room temperature. Importance of labeling: prevents confusion, improves traceability, ensures quality control, reduces laboratory errors, enhances safety. Hazard labels. Hazard labels indicate toxic, corrosive, flammable, oxidizing, biohazard. These symbols help laboratory personnel handle chemicals safely. Common labeling mistakes: missing concentration, illegible handwriting, wrong dates, missing hazard information, using labels that fall off. Good practices: use waterproof labels, write clearly, attach labels securely, replace damaged labels immediately. Viva questions. Why is labeling important? What information should every label contain? Why are hazard symbols necessary? Key point: a properly labeled solution prevents mistakes, improves safety, and ensures traceability. Storage. Correct storage of laboratory chemicals, reagents, and prepared solutions is essential for maintaining their quality, stability, and safety. Improper storage can cause chemical degradation, contamination, reduced effectiveness, and hazardous reactions. Storage conditions. Different chemicals require different storage environments. Room temperature, 20 to 25 degrees Celsius stable chemicals and salts. Refrigerator, 2 to 8 degrees Celsius enzymes, antibodies, and many biological reagents. Freezer, minus 20 degrees Celsius or minus 80 degrees Celsius. DNA, RNA, proteins, and long-term biological samples. Protection from light. Some chemicals are light-sensitive and degrade when exposed to sunlight or strong laboratory lighting. These should be stored in amber-colored bottles or dark cabinets. Examples: silver nitrate, vitamin solutions, certain antibiotics. Chemical compatibility. Never store incompatible chemicals together. For example, acids should be stored separately from bases. Oxidizing agents should be separated from flammable substances. Flammable chemicals should be kept in approved flammable storage cabinets. Inventory management. Laboratories should maintain an inventory system to track reagent quantities, monitor expiry dates, prevent unnecessary purchases, ensure first in, first out, FIFO, usage. Common storage mistakes: leaving bottle caps open, storing chemicals at incorrect temperatures, using expired reagents, failing to label storage containers, placing incompatible chemicals together. Best practices: always read the manufacturer's storage instructions. Keep storage areas clean and organized. Check expiry dates regularly. Record storage conditions in laboratory logs. Dispose of expired chemicals safely according to institutional guidelines. Viva questions. Why is proper storage important? Which materials should be refrigerated? Why should light-sensitive chemicals be stored in amber bottles? What is the purpose of FIFO in laboratory inventory? Key takeaway: proper storage preserves reagent quality, extends shelf life, ensures laboratory safety, and guarantees reliable experimental results. In the final section, I'll cover slides 16 to 20 with the same detailed level of explanation, including common mistakes, good laboratory practices, summary, quiz answers, and the concluding remarks. Common mistakes. Even experienced laboratory professionals can make mistakes if they do not follow proper procedures. Many laboratory errors are preventable and usually occur due to carelessness, lack of training, poor documentation, or failure to follow standard operating procedures, SOPs. These mistakes can lead to inaccurate results, contamination, wasted resources, equipment damage, and even serious safety hazards. 1. Incorrect measurements. One of the most common laboratory errors is inaccurate measurement of chemicals or liquids. Examples include reading the meniscus incorrectly, using uncalibrated equipment, incorrect pipetting technique, weighing chemicals without zeroing the balance. Prevention. Always calibrate instruments. Read the meniscus at eye level. Use analytical balances for precise weighing. Select the correct pipette for the required volume. 2. Contamination. Contamination occurs when unwanted substances enter a sample or solution. Sources of contamination include dirty glassware, unsterilized equipment, touching sterile pipette tips, leaving containers open, cross-contamination between samples. Consequences: false laboratory results, growth of unwanted microorganisms, failed experiments, sample rejection. Prevention: work aseptically, sterilize equipment before use, replace gloves when contaminated, keep containers closed when not in use. 3. Poor labeling. Improper labeling is a major cause of laboratory errors. Examples: missing concentration, no preparation date, wrong sample identification, illegible handwriting. Poor labeling can lead to sample mix-ups, incorrect diagnoses, repetition of experiments. Safety hazards. 4. Failure to follow SOPs. Ignoring laboratory protocols can result in experimental failure, safety incidents, poor reproducibility, invalid data. Every procedure should follow the approved SOP exactly. 5. Poor documentation. Laboratory notebooks should contain date, procedure, observations, calculations, results, Signature. Incomplete records make it difficult to repeat experiments or investigate errors. Practical example. Suppose a technician accidentally uses 1 mHCl instead of 0.1 mHCl during a titration. The result will be completely inaccurate, requiring the experiment to be repeated. This wastes chemicals, time, and resources. Ways to avoid laboratory mistakes. Read SOP before starting. Check calculations twice. Label everything immediately. Maintain a clean workspace. Use calibrated instruments. Record observations promptly. Ask for guidance if uncertain. Good laboratory practices, GLP. Good laboratory practices, GLP, are internationally recognized quality standards designed to ensure laboratory work is conducted safely, consistently, and accurately. GLP promotes reliable data generation, protects laboratory personnel, and ensures compliance with regulatory requirements. Objectives of GLP. Improve laboratory quality. Ensure accurate and reliable results. Protect laboratory personnel. Maintain scientific integrity. Enhance reproducibility. Principles of good laboratory practice. 1. Read SOPs before starting. Never begin an experiment without reading the complete procedure. Understand. Objectives. Materials required. Safety precautions. Expected outcomes. 2. Maintain proper documentation. Every observation should be recorded immediately. Good documentation includes date, time, sample ID, instrument used, observations, calculations, final results. Remember, if it isn't documented, it wasn't done. 3. Maintain cleanliness. A clean laboratory reduces contamination. Always clean benches before and after work. Wash glassware properly. Dispose of waste immediately. Return equipment to its designated place. 4. Dispose of waste properly. Laboratory waste should be segregated into biological waste, chemical waste, sharps, general waste. Improper disposal can harm people and the environment. 5. Equipment maintenance. Laboratory equipment should be cleaned regularly, calibrated, serviced, checked before use. 6. Quality control. Always include positive controls, negative controls, calibration standards. Duplicate samples. These help verify the reliability of experimental results. Benefits of GLP. High-quality data. Reduced errors. Improved laboratory efficiency. Better safety. Compliance with accreditation standards. Viva questions. What does GLP stand for? Why is documentation important? What types of laboratory waste exist? Why should equipment be calibrated regularly? Key takeaway. Good laboratory practices are the foundation of safe, accurate, reliable, and internationally accepted laboratory work. Summary. In today's lecture, we covered the essential principles of laboratory protocols and solution preparation. Let's briefly review the major concepts. We learned that laboratory protocols are standardized written instructions that ensure experiments are performed consistently. Following SOPs reduces errors and improves reproducibility. Laboratory safety is essential and includes wearing PPE, understanding hazards, and following emergency procedures. Different laboratory solutions include stock solutions, working solutions, standard solutions, buffer solutions. Every solution consists of solute, solvent. Concentration can be expressed as percentage, molarity, normality, ppm. Molarity is calculated using M equals moles/liter. Dilution is calculated using CD equals C₂V₂. Buffers maintain stable pH. pH meters provide accurate pH measurements after proper calibration. Correct labeling and storage improve laboratory safety and quality. Good laboratory practices improve documentation, safety, and experimental reliability. Overall message: successful laboratory work depends on accuracy, precision, safety, documentation, quality control, professional ethics. Remember, good science begins with good laboratory practice. Viva questions. Define laboratory protocol. What is molarity? Why are buffers important? State the dilution equation. What are good laboratory practices? Key takeaway: safety plus accuracy plus quality equals successful laboratory science. Quiz. This quiz is designed to assess your understanding of today's lecture. Question 1. What is a laboratory protocol? Answer: a laboratory protocol is a standardized written procedure that provides step-by-step instructions for performing laboratory experiments accurately, safely, and consistently. Question 2. What is molarity? Answer: molarity is the number of moles of solute present in 1 liter of solution. Formula: M equals moles/liter. Additional practice questions. 1. Define a solution. 2. Differentiate between stock and working solutions. 3. State the dilution formula. 4. What is the function of a buffer? 5. Why is pH important? 6. What information should appear on a laboratory label? 7. Why should chemicals be stored properly? 8. What are good laboratory practices? 9. What causes contamination? 10. Name 5 pieces of laboratory equipment. Activity: ask students to calculate molarity. Perform a dilution calculation. Identify laboratory equipment. Explain hazard symbols. Prepare a correctly labeled solution. Key takeaway: continuous practice and regular review strengthen laboratory knowledge and practical skills. Thank you. Thank you for your attention and active participation throughout today's lecture. Today, we explored the importance of laboratory protocols, solution preparation, laboratory safety, concentration calculations, dilution techniques, buffer systems, pH measurement, proper labeling, storage, and good laboratory practices. These concepts form the foundation of laboratory work in medicine, pharmacy, biotechnology, microbiology, biochemistry, pathology, nursing, and many other scientific disciplines. As future healthcare professionals and scientists, always remember that accuracy is more important than speed. Safety is everyone's responsibility. Proper documentation ensures reliability. Following protocols produces reproducible and trustworthy results. Continuous learning improves laboratory skills. I encourage all of you to apply these principles during practical sessions and clinical laboratory work. Mastering these fundamentals will help you become competent, responsible, and confident laboratory professionals. Final message: a successful experiment is not just about obtaining results, it is about obtaining accurate, reliable, safe, and reproducible results through proper laboratory practices. Thank you. Any questions?